Can you guys see my screen?
Yep, perfect.
Great. Well, thanks very much, Nargis, for the invitation. And for my colleagues and friends at NYU, I owe you a visit in person. And if you would have me back sometime next year, I'd love to come in person. So my talk today will be, have as a backdrop a lot of the progress that we've been seeing in the AI field over the past several years, much of it, a lot of it by our colleagues at NYU. So for example, progress in computer vision, such as object recognition, progress in reinforcement learning, such as learning to play games, progress in learning how to understand natural language and ask and answer questions that are posed in natural language, and questions about learning representations of data that would then allow one to create new synthetic data. So you see in the bottom right an example where on the far left side and far right side are actual people's faces, and one can sort of move a knob to generate synthetic faces that look anywhere in between those two real ones and look very realistic. And so what my talk about today is how some of these advances in the broader AI field translate into the pharmaceutical industry, could potentially translate into the pharmaceutical industry and into clinical research with regards to real-world data. And what makes this translation particularly feasible is because the democratization of machine learning that has been happening in all of these other fields due to, for example, large data sets, very cheap batch processing, and high quality open source software has just made it much easier to translate advances and has led to a much faster pace of research in the field. So specifically, when one thinks about the healthcare industry and where real world data comes from, it comes from a variety of places. Of course, you have electronic health record or EHR systems that generate in which you have data recorded, such as notes, labs, imaging data from providers. You have data sent to payers, health insurance companies. That claims data contains a wealth of information about what's happened to patients and what treatments they're on. You have disease registries that patients are in that track patients with particular chronic diseases, perhaps even rare diseases where any one institution may not have enough data. And then on the pharmaceutical industry side, you have these clinical trial management systems with a wealth of data from randomized control trials that have been done for new drugs. And the data that one collects just as part of this practice of medicine and practice of bringing new drugs to the market could be really valuable for a number of downstream use cases, ranging from thinking about new ways of doing regulatory approval, for example, label expansions from off-label use, single-arm experimental trials, to drug development, discovery of novel disease subtypes, and ideas about how one could come up with new drug targets based on each of those subtypes, clinical trial design, and ultimately clinical care as well. And so my talk today is going to touch on several of these different buckets. And just to kick things off, I'll give a very simple example of how one could use data from randomized control trials to think about how to do better patient selection, either for the next phase of a study or for a related drug. And so this is work that my lab, in particular, a postdoctoral fellow of mine, Frederick Johansson, did several years ago in collaboration with Brigham and Women's Hospital and with Genentech. And we were asking for which patients with rheumatoid arthritis are they most likely to achieve remission with tocilizumab monotherapy, which is one of the drugs created by Genentech. We took data from 4 different randomized controlled trials. Each of those randomized controlled trials had a number of arms, and we took data just from the monotherapy arm for the study. And altogether, when combining them, you had over 1,000 patients. And then we created a number of features that one could derive from the baseline data in each one of the studies. Things like past drug exposures, baseline CDAI score, which measures disease activity, a number of measures derived from biomarkers, and even geographic information and demographics. And we use that to try to learn a predictive model of which patients are likely to to achieve remission with this drug. And we found that using that model, you could achieve a what's called an area under the receiver operator characteristic curve, which is a measure of predictive performance, a very crude one, of 0.756, and that should be compared to random guessing, which would be 0.5. And what's particularly interesting is if you think about how one might then try to deploy those models, one could try to find, for example, thresholds for which the negative predictive value is very high. Similarly, you could think about finding subsets where the positive predictive value is very high. And each of those are examples of, can you find cohorts of patients that are for sure going to respond or for sure not going to respond? And then thinking about then using that to guide whether patients should receive these treatments or perhaps just immediately jump to other treatments. Now, that study was done with data from randomized control trials. And randomized control trials, as I'm showing on the right-hand side here, have really nice data typically. So very well-defined time points where patients will receive treatment, and for at which point we will have documentation of what happened, their existing symptoms, comorbidities, and so on. So not that much missing data in this setting. And that should be contrasted to the data much of the data that I was alluding to in the first part of the talk, which is real-world data coming from just the practice of medicine, right? So if a patient is not going to any healthcare facility, typically we don't have much data on them. When they're hospitalized, we suddenly get a wealth of very rich data on those patients, whether it be from clinical notes or physiological data or lab test results. And then a long time might go on until you have more observations for these patients. And so real-world data is very rich. It has a potential for giving us insight on, for example, all 300+ million people in the United States, even if they're not in clinical trials, but it has a number of challenges one has to address in order to get signal from the data. And so I'll go into much more detail into some of those challenges and some solutions towards them in the rest of the talk today. So the first part is going to ask, could we learn patient simulators from real-world data that arises from electronic medical records, but also data that might come from randomized controlled trials? And this is work that was led by my current PhD student, Hussain, who is an MD PhD student at MIT, and a former student of mine, Rahul Krishnan, who used to be at NYU and is now a professor at University of Toronto. So the key idea is to think about the following journey of a patient with chronic disease, and then to think about how one could try to take data up until one point in the journey and then try to predict what's going to happen to the patient for the rest of the journey. So what you're seeing here is a plot, a sketch. X-axis is time, y-axis is disease burden. The disease burden might be increasing in this chronic disease until the time of diagnosis. Patient might get a first-line therapy. Maybe that actually decreases the disease burden. Eventually, they might have a relapse. You see that a lot in cancer. They might get a second-line therapy, and so on. Now, if you start looking at the data in a little bit more detail, and you look at what data you get at each step of that process through a patient's chronic disease trajectory, you'll have, for each treatment line, you'll have information on what therapy was provided. Very often, patients are taking more than one treatment at a time, so you can think about combination therapies that they're receiving. You have biomarker data that might be observed across time. These might be coming from laboratory test results. You might also have other data that you would derive directly from clinical notes, such as symptoms or complications of treatment or the disease. And you also have a lot of baseline data. I use the word statistics here, but it's really more about baseline data. So you might have the patients, if it's a patient with cancer, maybe you have some genomic data from the tumor itself, but either at baseline or maybe also at later disease stages. And what I'm showing in orange here is that there might be a lot of missing data. So for example, if the patient actually didn't visit the clinic at any point in time, then you don't really know what their laboratory test results are at that point in time. And for any one lab test as well, you might not have that observed at any one particular visit for a variety of clinical reasons. So zooming out to outer space, there are several questions that we might be interested in asking about how a patient's disease progresses across time. For example, you might take the data you have available at the time of baseline. So for example, at the time of diagnosis, but before initiating treatment. And if we had a patient simulator in this dreamlike world, we would be able to take that baseline data and predict all of the patient's future data for the next few years. So predict, for example, all of the future biomarkers that might be observed for that patient as their disease progresses across time. And if we could do that simulation, then we could just read out from the resulting rollout many of the information that would be useful for clinical care. So for example, we could just read out from that simulation a survival time, and that would give us a prediction of survival time or time to progression. We could also get a personalized side effect profile by seeing, okay, well, if there's a patient who's having calcium levels rise up, maybe they're likely to have a particular complication. If you see symptoms that show up at 10 months, then we would have an idea of, well, maybe those symptoms are due to the proposed treatment, and so on. A second example might be, well, now the patient's undergone therapy. We have, let's say, 6 months of therapy. We observed some measurements for them. And now we want to simulate the next 6 months of data for this patient. And if we could do that, then it might be useful, for example, to predict non-response to treatment, so that we could very quickly recognize that the patient is not responding to treatment, stop it, switch to a different one. Or likewise, maybe the patient has reached remission, and we would now be predicting that the patient might be having a recurrence of the disease. So you might want to initiate treatment very quickly. And so you can think about this as one way of constructing synthetic biomarkers that give you insight into what's going to happen to the patient in the future and allow you to act much quicker. Now, although I hid the detail from you up until now, those of you who have thought about causal inference might recognize there's something very odd about what I'm showing you here, because I'm actually conditioning on treatment, or I'm generating treatment as well. And that leads me to the third example of what you might want to do with these patient simulators, which is you might want to propose a candidate treatment regime. This might be a dynamic treatment regime, for example, and then ask, well, given the baseline data we have in the patient and a proposed treatment regime, can we predict what's going to happen to a patient over the course of their disease? And then, of course, if you could do that well, then you could use that, you could try different counterfactual treatment regimes and use that to guide choice of treatment, which is really the ultimate goal of precision medicine. So I've spoken up until now about what I mean by patient simulator, but now let's think a bit about how we might try to learn these from data. So the naive approach for trying to learn this, if you have a machine learning hat on, as many people in, for example, the Courant Institute have been working on, is you'd say, okay, this looks like a time series data, and we know of good ways in machine learning to model sequential data, such as using an autoregressive model, like a recurrent neural network. And those are very commonly used in the natural language processing community. So you might condition on the patient's data up until some point in time and learn a model that always predicts the next point in time's data, their full data. And then if you could do that, then you can then condition on the next point of time's data and all of the previous data and predict the subsequent point in time's data. And in that way, roll out and simulate all of the patient's future history. And so this is, just to make the analogy very concrete, and to give you some intuition on how this works in the natural language processing community, consider the following sentence. Diego fell off his bike and began to blank. Before you guess what the blank is, let me give you a bit more context. Diego is my 6-year-old son. Maybe all of you could just open up your chat on the very bottom and type in what you think that blank word is. As far as I can tell, the chat is open. Okay, you know what? Now I realize I think the chat is not open.
Oh, I'm sorry. Yes. While I will enable it, give me a second.
Okay.
We actually have one question while we enable it from Jennifer in the question and answering. While you answer that, I'll enable this.
Yeah. So I can read it out loud. So Jennifer, thank you for the question. Since the predictions are only as good as the data being fed into the model and the model itself, is it ethical to manage treatment based on predictions? Well, naive predictions, definitely not. And part of what makes this machine learning healthcare field so interesting and challenging today is we are working as a community to develop checks and balances for how one can sanity check that what the models are predicting are accurate, that they are not biased, and that they will continue to work even under dataset shift, among many other challenges. So just to be very clear, what I'm proposing at this point in time is a vision for what we can do as a community over the next several years with many of those checks and balances built in. And so thank you, Nargis, for enabling the chat. And so we see answers like cry, cry, bleed, bleed, cry, 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 bleed. So something like 80% likelihood that the answer will be cry, and 20% likelihood the answer will be bleed. And the last time I did this experiment with an audience, it turned out that the 80% cry came from a population of people who had children, and the 20% bleed were from people who didn't have children. Just to give you some insight into the prior knowledge that goes into these predictions. So, but generally speaking, my answer would have been cry. And we can see that all of us had a pretty good idea of what the next word is. And we can do that because we have a very good understanding of two things. We have a good understanding of language and what types, when a verb will follow a noun, when you would expect to see a preposition. And we also have a very good understanding of the world. And so I told you that Diego was my son. You know that falling off a bike can hurt. And so putting those two pieces together, your prior knowledge about the world and your knowledge about language, makes it very easy to predict that the next word should be cry. And that's exactly what these autoregressive language models do. They condition, they're trained for exactly this type of task, given any sequence of words, try to predict what the next word is. And again, the analogy I'm trying to make is that one can imagine trying to use exactly the same techniques here. Except there are a few differences, and those differences end up making those approaches not work as well as one might hope. One difference is that instead of outputting a single word, you're outputting many dimensions. For example, in some results I'll show you later, we're outputting 16 biomarkers at every time point, but that could have easily been hundreds or thousands or millions. Time intervals are irregular. There's a lot of missing data, as I alluded to, and we're interested often in simulating these counterfactuals of what would happen under one treatment versus another, whereas there's no notion of a control signal in the typical language prediction problem.
David, there's a question from Ernest Davis.
Hey, Ernie, let me see if I can find your question. One moment.
It's in the chat. I can repeat
I see it now. Do I really want to claim that autoregressive models have knowledge about the world? Yes, I do. Ernie, I'd love to chat with you more about that later. But I think we're seeing that a lot now with large language models, where I do believe that encoded in those billions of parameters is a huge amount of world knowledge. But that's a topic for a much broader discussion. I'd love to have that with you later. So thank you, Nargis, for pointing that out. So, and the last point I want to make here is that there's very little data to learn from. So, many of these data sets might contain just several thousand patients, as opposed to the millions or billions of sentences that one can imagine learning from in a natural language context. And as a result, recurrent neural networks tend to overfit quite a bit on these types of tasks. So there's been a lot of prior work on this idea of building a patient simulator. And one very nice example comes from work by Suchisaria and her student Peter at Johns Hopkins, where they looked at an example of a patient with some lung capacity issue. So maybe an asthmatic patient or a patient with COPD. And they asked, how could one try to model the lung capacity as a function of time and as a function of interventions that might have been received? So the x-axis in this plot is time, y-axis is lung capacity. On the very bottom, I'm indicating what treatments the person received. So the blue drug or the green drug might have been in their history, and the black dots are the actual observed data for the patient's lung capacity in that historical window. And so their approach would builds on what are called Gaussian processes and shows how one can condition on treatment information, which then, when learned in the right way, gives you what they call counterfactual Gaussian processes. And then one could use those models for rollout. So for example, here, this blue line is suggesting what would happen to the patient's future lung capacity if they did not receive any future treatment. That's what the not sign here next to the Y indicates. And you might ask a different question. Well, what happens if they receive the green treatment at time step 6? And then there, the prediction might be this orange line. Or you might ask, what happens if they receive the blue treatment at 2 different time points? And then their prediction might be this purple line. And although it's beyond the scope of the talk to explain Gaussian processes and how these work, intuitively, this is something like line fitting, but in a highly nonlinear and much more clever way. So, right, if you look at this history over here and you see these dots that are going down, it might be reasonable to expect that the predictions, the future dots, are going to still be going in the downward direction. And they show how to fit nonlinear models that can do similar extrapolations. So this type of approach has a lot of really nice properties to it, but it also has a few limitations. For example, it's designed to model single biomarker across time. It has very limited ability to condition on baseline information. I had alluded to how we'd really like to be able to condition on all of the patient's genomic observations, for example, at baseline, and use that to get more accurate predictions of future progression. And the treatment response functions are assumed to be additive, which is not a big limitation. It's not clear that that should hold true in many real-world settings. And so the approach that my students and I have been working on for several years now, it goes by the name of deep Markov models. And at a high level, it's something that many of you, I think, would have already been familiar with. So the notion of hidden Markov models shows up a lot in machine learning, in the pharmaceutical industry. And the general idea there is you have some hidden state that's evolving with time. For example, the patient's disease state, ZT, is evolving with time, but it's not assumed to be known. And rather, what you observe are a number of observations, which I'm denoting here as the X's. These might be, for example, laboratory test results if they're observed, symptoms, maybe even genomic data. And there are a number of actions that are performed, which are shown in these orange circles here. These actions could be treatments. And the arrows denote purported effects. So for example, the model is assuming that the patient's state of the next time step, Z2, for example, is a function of the patient's state of the previous time step, Z1. And that the action of the previous time step, U1, affects the action of the next, affects the patient's state of the next time step, Z2, and so on. Now, what's different about our approach is that we're not going to assume that the patient's state, their true underlying which we want to mean their true underlying physiological state, we're not going to assume that that has any simple form. Rather, we're going to assume that it lives in some higher dimensional continuous space, such as a 100-dimensional real valued number. And we're also not going to assume that the transition distributions between the patient's state and actions and the effect that has on the patient's state of the next time step, we're not going to assume that that has some simple form. We're going to assume that it's highly nonlinear, and we're going to try to learn those relationships from data. And if we could do so, then this would provide an in silico model for assessing the effect of interventions, these actions, by forward sampling in the model in the way I alluded to earlier when I spoke about patient simulators. So in our work, so this line of work started in 2017, and it was really one of the first papers to show how to bring together probabilistic graphical models and deep learning methods. And in particular, we parameterized the conditional distributions of the Z's at the next time step, conditioned on the Z's and the U's of the previous time step, using deep neural networks, as I'm sketching on the right-hand side here. However, although we found that we got reasonable results in one health setting and very good results outside of healthcare setting, as we went to try to apply this to more and more health data sets, we realized that these methods still overfit more than we would like them to. And so
David,
the next,
there's a question, sorry for interruption. There's a question from Mingju Fu, might be very related in the
sure, let
Q&A.
me give me 1 second to try to find it.
In the
I see
Q&A.
it. Thank you. Yeah. So there's a question about feature selection. When you have limited samples but tons of features, how do you do feature selection? Do you do feature selection first before putting the features in the model? Thank you for your question. So I'll just go back 1 slide to here. So I spoke about how we're treating these Z's as living in 100-dimensional space, and these X's could also be very high dimensional. And certainly we have a large number of parameters in this deep neural network going from Z's and U's to Z's. And you might very much worry about overfitting here. And so there are ways to try to prevent yourself from overfitting, such as feature selection. You can imagine feature selection happening in the X space by excluding particular symptoms or lab tests from your model and just modeling ones that you think are clinically relevant. But a major challenge there is that you want to be able to discover new things. So you'd like to be able to recognize that some other biomarkers actually might give you insight into the patient's future disease progression, even if that hadn't been previously known. So we typically don't want to exclude too much at the start when we try to do these models. And instead, we try to prevent ourselves from overfitting by using a technique in machine learning that goes by the name of regularization. And regularization, when you're learning deep Markov models, could take several forms. It could be in the form of weight decay, where we ask that no one weight gets too large weight value. It could be in the form of early stopping, where you choose an epoch to stop learning at, depending on your validate performance. Okay, so this gets me to my next theme, which is what are other ways to try to limit the model capacity, yet still allow for discovery of the nature I mentioned earlier. And what we came up with over the last couple of years is a method to build some domain knowledge into how treatment effects manifest. And particularly, we were inspired by the pharmacodynamic and pharmacokinetics literature coming from the pharmaceutical industry, which very often thinks about how a drug would affect a particular biomarker or physiological state as a function of time once you initiate that treatment. So for example, here you might see that the blue line might be your assumption of what would happen in the absence of treatment, and the red line might be what you might expect to happen if there was treatment. So maybe you would see some drop in disease burden or some other measure, and then a gradual incline again. And so there are all these different differential equation models that the community had developed for how to model pharmacodynamics in a realistic way for various different types of chronic diseases and different types of treatments. And what we've been asking is how we could try to bring those insights into our deep Markov model. And the first thing that we propose doing is to, instead of modeling on just the biomarker space, which is what the traditional PKPD approaches do. So traditional PKPD approaches will just typically model the effect of a single intervention on a single biomarker, whereas we're interested in this much more ambitious setting of modeling lots of biomarkers with many interventions. And so the way that we tackle that is we look at the response in latent space. So instead of assuming that we're going to look at the effect on one biomarker at a time, instead we're going to model, now you see on the left-hand side here that I have a Z here. We look at the effect of the treatment on each of those individual 100-dimensional latent dimensions. And but then we still try to use as much of the prior knowledge of these pharmacodynamics as possible when parameterizing that transition distribution. And so the approach that we come up with, we call the state space model, SSM PKPD. And what we modify is that transition distribution. And we introduce several different, what we call PKPD effect functions, which have very simple parametric forms. And then we learn how to modulate the effect of them, in essence, figuring out for each patient at each point in time, which of these PKPD mechanisms is the best fit for making a prediction of the treatment effect for the next time step. So for example, one of those functions would take the following form, where the mean of the Z at the next time step is given to you by the Z of the previous time step times 1 minus rho times log of the Z of the previous time step squared, and so on. And if you were to actually plot that, you would get these plots that you see on the left-hand side. And this would be an example of how you would model slow, gradual relapse after treatment. So we have a whole library of these different functions pulled from the literature that we then allow our learning algorithm to choose from as it proceeds. So we tested this on a data set of 1,000 patients with multiple myeloma. The data set is publicly available, provided by the Multiple Myeloma Research Foundation. And if you look up our paper, you'll see a link to a code base that allows you to reproduce all of the results I'll show you here, reproduce the results on this publicly available data set. And I would love to see more people in the community start testing out disease progression modeling ideas on this publicly available data set that I think is very rich. So here we worked with an oncologist to select 16 clinical biomarkers to model across time. And we looked at 9 indicators of treatment, given that there were many different treatments that were being considered for this patient population. And in this work, we used 16 baseline features, so still relatively low dimensional, of different projections of genomic data. So here's an example of what I mean when I talk about simulating the effect, the progression after some period of time. The x-axis in each of these plots is time, but measured in 2-month intervals. So roughly 9 months in, roughly 9 time points in corresponds to 18 months of the patient's trajectory from the time of starting treatment. And I'm showing you here 2 plots. Remember that I said we were modeling 16 biomarkers across time? Well, here are just 2 of those 16 biomarkers, but we're actually providing simulations for all 16 jointly. These 2 biomarkers are very important for understanding the patient's disease burden in multiple myeloma. The actual data for the patient is shown in these blue dots as a function of time. And what you'll notice is that our predictions perfectly align with observed data up until nine time points, and that's because we're conditioning on 9 time points. So this is an example where we take data up until 18 months, and then we predict what's going to happen after those 18 months. And here I'm showing you 2 different sets of predictions. The first prediction shown in red is given to you by a state space model, but where we're using a linear transition distribution. So we're not allowing for nonlinearity in the latent space. In particular, we're not allowing for nonlinear relationships between treatments, previous disease state, and next disease state. And in gray here, I'm showing you an example of the predictions from our PK/PD model. And I should mention that this patient is chosen from our held out set. So this patient's data was not used during training. And what you see is that the predictions align much better with the actual observed data, what happened to a patient. So these gray dots are relatively close to these blue dots, which gives us hope that we're actually learning something reasonable here. Now, a different way to try to measure this quantitatively would be to look at what's called the held out likelihood or perplexity of the observed data under different types of models. And this, in essence, is a metric that quantifies how well the model predicts the sequence. The lower the number, the less surprised the model is by the next observation, right? Think about when I asked you to fill in the next word and all of you say cry and bleed. If I had told you the next word was smile, then you would have been really surprised and this would be a very high value. Here we're looking for, so we want the numbers to be low, which would be less, the model is less surprised. And what we find is that in comparing a number of different models, including that SSM linear model I spoke to you about earlier, but also models that use more naive ways of introducing nonlinearities and recurrent neural networks that don't make any assumptions about conditional independence properties, we find that the SSM PK/PD model actually gets substantially lower substantially higher held out likelihood, lower negative likelihood. So
yeah. David, interrupting you, there's a question also by Nan Wu in the Q&A.
Thank you.
Before we move on.
Okay, I see the question. For such a model using multiple biomarkers and patient information, is there any analysis on explainability, such as what are the features that it relies on? And then also asks, well, how important do I think that is in terms of application? It's a great question. In our paper, which I'm happy to send you these slides later, and I think you said the video will be recorded, and the link to this is from my website, or you could just look up Zishan Hussain on Google, and you'll find the paper. You'll see in the supplement some attempt at an interpretability analysis. And I think one of the nice things about this approach is that these mechanisms actually provide new opportunities for doing that interpretability analysis. For example, though I'm not showing it to you here, each of these OSO conditions on the baseline data, and you can look to see how does the baseline data affect which of these mechanisms is chosen at each point in time, and what exactly is treatment doing. And there's certainly a lot of room for more work on that direction, and I do think it's very important because here we've done a test on one data set, but what you're ultimately interested in is if we were to deploy this model in a setting which is a little bit different from what we were trained on, how would it do? And that's not something you can necessarily evaluate if you don't have data that's fully representative of what you should expect to see at test time. And so these types of interpretability analyses really allow domain experts who have lots of insight into how the data might be shifting from one patient population to another, or from one geographic location to another, how that might actually impact the model's predictions. Okay, so the second part of the talk today, and the second and third sections are much shorter, looks at a different question of how one could discover disease subtypes. And this is work that was led by my PhD students, Irene Chen and Rahul Krishnan. I already mentioned Rahul, but Irene graduated last year, and she is currently a postdoc in Microsoft Research and will be starting as an assistant professor at UC Berkeley and UCSF next year. So many different diseases, despite having a common name, actually have quite a bit of heterogeneity. There was a really nice paper several years ago in the asthma community showing how to use a k-means clustering algorithm to discover disease subtypes. There was follow-up work by Finali Doshi and many others, which looked at how to do that in a data-driven way for autism. And there is, of course, a very well-known characterization of different subtypes in heart failure, such as HFpEF and heart failure with and REF. So here, what we're looking for is, can we develop an algorithm to find disease subtypes automatically, where intuitively a subtype corresponds to similar patients who might respond similarly to treatment. And if we could find those subtypes, then it might be useful for drug discovery efforts, because you might be able to recognize that patients in different subtypes might have different possible targets for treatment. Now, the key limitation which this work focuses on is that we want to be able to account for systemic biases in the data that we use for the subtyping analysis. And this is really important when it comes to real-world data, because how patients enter into the data set really affects the conclusions you can draw. So consider the following example. Here we have three patients, patients A, B, and C. And in this idealized example, I'm going to conjecture that we have some idea of how to stage patients. So suppose we knew that there is a biomarker, and that very light colored values of that biomarker means the patient has a very mild disease, very dark colored means the patient has a very severe disease. Now, here you might see these three patients, and given what I just told you, you might say that patients A and B look very similar to another. They seem to progress through their disease at very similar rates, whereas patient C looks very different from patients A and B. Now, imagine that you had data that looked more like this. So here you have left censoring. Patient A might not have entered the data set until the point denoted by this square bracket. Patient B entered the data set over here. Patient C entered the data set over here. Now, this can happen for a variety of reasons in the healthcare system, but one example has to do with access to care. Patients who live near academic medical centers or are wealthy enough to pay for frequent medical visits might have their diseases diagnosed much earlier than other patients who don't have as much access to care. As a result, you might find that you get data from much earlier in their disease trajectory, patient B compared to patient A, for example. Now, we don't want that censorship to actually affect the results of the subtyping we do. And so the name of the game here is to figure out how we can still discover subtypes despite the fact that we have this left censorship, which very much affects the disease stage the patient enters with. Now, there are a couple of obvious approaches to try to deal with this problem. Option one, for example, might be to say, let's try to manually realign the data. So if you had a good idea of how to stage patients, for example, a good idea of what a biomarker of severity would be, then you could imagine trying to shift patients' disease trajectories to try to get them to all start at very similar stages of their disease. But the challenge there is that that's very time consuming, and perhaps even more importantly, even to do that assumes that you have a good way of doing staging. But what happens if we don't yet have a good way of doing staging in this disease, or we don't believe the current instruments that are used for staging? A second approach might be to just say, okay, well, let's just ignore the alignment issue and just try to do clustering without that. And that has other failure points. So here I'm showing you exactly the same data that I showed you over here, but I've just shifted all of the timelines to start at the left bracket. And if you're to try clustering this data, you might incorrectly conclude that patients A and C are similar when indeed they're actually quite different. So the approach that we propose is to develop a statistical model that at the same time tries to assess the patient's disease stage and their subtype. And in doing so and trying to learn that model, explicitly disentangle between subtype identity and the alignment between these trajectories. Of course, we also want to be able to deal with the fact that we have multivariate time series where we're not just modeling a single biomarker across time, but a large number of biomarkers, and there also might be a lot of missing data. So this approach goes by the name of Sublime, and the intuition behind what it's doing is that we're modeling the latent structure for the patient. The Z is going to characterize their disease subtype. We're modeling their alignment time, delta, at the time of entering the data set. So delta denotes their disease stage, for example. And we're using those two latent variables. We're conditioning on them to say that, well, conditioned on knowing the patient's disease type and their alignment, then that together will provide us a way of modeling the biomarkers you would see at any point in time in the data set. A very important question to ask in this type of work is this even possible? Like, can you actually distinguish between disease subtype and disease stage? And so part of our paper also explores the identifiability question, which is a very statistical question. Although I won't go into this talk, I encourage you to look at that. So at a very high level, the way our approach works is very, very similar to the deep Markov model that I showed you earlier. We're going to again assume that we can describe the patient's underlying physiology with some variable, some latent variable, and some high dimensional continuous space. That's what we're calling Z here. The only difference is we're not going to model Z as a function of time in this work. We're going to assume there's a fixed Z at one point in time, such as the time of entry into the data set, but that's shared throughout the whole entire sequence. We're also going to assume that we have an alignment value, and that alignment value is going to be what modulates the Z to say, okay, Z plus this stage of the disease then results in some observations that you would see with patients' biomarkers. And so we then develop a model which combines Z and delta to describe the likelihood of any sequence of observations Y are the patient's biomarkers, as a function of the times and the time intervals that those biomarkers were observed. Those are the X's. So although I won't go into the details, the way that we do that parameterization is actually using polynomial functions. And the degree of the polynomial actually ends up playing a big role in the identifiability arguments that I alluded to earlier. So to evaluate this is actually very complicated because we don't have many examples of data sets where we have a notion of ground truth, disease stage, and subtyping for patients. So we're going to evaluate first with synthetic data and then with a real-world data set where we have some idea of what to expect just from basic clinical knowledge. Now, to measure clustering performance, there's a bit of a subtlety here. If you look at the far right-hand side, I'm assuming that there are 3 clusters here. I'm showing you the colors denote the true clusters. And suppose that our algorithm were to output a clustering shown on the far right side, right? So you have the data points that were all yellow before are all labeled cyan now. The data points that were labeled that in the true clustering is purple are now labeled yellow, and so on. Now, clearly, this model has actually discovered the correct clusters. They just have different names. And so we need a metric to measure the performance of clustering that's invariant to just relabeling the clusters. And that's what the adjusted RAND index or ARI quantifies. And so a high value of ARI means even after finding and relabeling, we're getting how well we're doing at matching the true clusters. The second metric that we're interested in measuring is how well we're recovering the ordering between patients at different stages of their disease. So the way that we could do that, at least with synthetic data, is we will have some ground truth values of delta, which is in essence, how much left censorship was there truly for a patient. And we'll compare the inferred deltas to the true deltas. And we can measure how well we predicted the deltas by using, for example, the Pearson correlation between the true and predicted deltas. You want that to be as high as possible, or by a swaps metric, which cares a little bit less about calibration and just asks, are we ordering the patients correctly? And there you want the number of swaps you would need to get to the correct order to be as low as possible. So we looked at a couple of different synthetic settings where we had the notion of ground truth to compare how our approach does in recovering that ground truth compared to other methods. So Sublime does very well at recovering clusters. So it has a value close to 1, which would be perfect. And it also does a very good job at getting the correct ordering between patients. You see that because it has a very low value of the swaps number and a very high value of Pearson correlation. Now you can try to do an ablation where you remove the delta, the alignment part of our generative model, and rerun the learning algorithm. It'll no longer be able to sort patients by their disease stage, but it will be able to cluster. And so you could ask then, well, how important was the disease staging component in coming up with the right clusters? And you see here that the cluster performance actually suffers quite a bit if you don't model the disease stage at the same time. You could also compare it to just doing a k-means clustering, using the raw sequences, and maybe even using some clever time warping between sequences. And that does quite poorly. You could compare it to other Bayesian approaches to this problem, and also approaches that are inspired by cross-sectional, by disease, but by alignment for cross-sectional data, such as you often find in the single-cell sequencing literature. And all of these other approaches perform very poorly, unfortunately. Okay, so that's with synthetic data. We next move to a real-world data set of 1,500 patients with heart failure who are all seen at the emergency department at Beth Israel Deaconess Medical Center at some point in time. And this is our collaborator, Stephen Horn at Beth Israel, who we worked with on this. And although these patients were all seen in the emergency department, we then get their data both before and after. So the inclusion criteria, having been seen in that ED, obviously leads to a lot of bias in which patient populations we're looking at. And the key question that we asked here is, if we were to try to do this type of subtyping, could we discover well-known subtypes of heart failure from this very biased real-world data? And to do the learning, we only used 12 features, all derived from echocardiograms across time for these patients. And we did not use any other side information from the electronic medical record, but we did use that side information to validate the subtypes. So we looked at a variety of different demographic and diagnosis codes after the fact, is to try to make sense of the clusters and see, is it discovering something actually meaningful? And so here I'm showing you the results. The algorithm has a final step which returns a total number of clusters that I won't walk you through, but here it finds 3 clusters, A, B, and C, all with roughly equal sizes. And then we could look to see which features derived from the side information not used during learning, but which we do have available, which features are statistically significantly different across these clusters as a way of trying to make sense of them.
David, there are question before we move further.
Yeah, actually, if you don't mind, I'm going to hold on for just a minute. I'll take that question in 1 minute. Actually, I'll take the question at the very end because I'm close to finishing. So here we see here that cluster B has patients from both diastolic and systolic heart failure where cluster A seems to have just patients with diastolic heart failure, and cluster C seems to have patients just with systolic heart failure. And so cluster B is actually quite an interesting one, and further investigation might want to dig in to see who are these patients that really are a mix of both diastolic and systolic heart failure. What differentiates them? You can see, for example, that they are many more women than men, and that they are tend to be more obese. And there's some literature that one can dig into to learn more. Okay, so the last piece of the talk, and I'll finish in the next 3 minutes. I want to bump up a level. We've been talking up until now about a couple of different ways that one could use real-world data, both for predicting disease progression and treatment responses, and for discovering disease subtypes from possibly very biased data. But in all of those cases, they relied on having data at scale for patients of a particular chronic disease. And a big problem we have is that getting that data is very, very expensive. In particular, it's very hard to get high quality data on symptoms, comorbidities, and other data that might otherwise be recorded only in clinical notes. And so over the past few years with my PhD student, Monica Agrawal, our collaborator who does human-computer interaction, Luke Murray, and a master's student, Divya Gopinath, we've been rethinking the whole process of clinical documentation from bottom up, with a lens on how we can really enable the future of real-world evidence studies. So we would like to be able to abstract from clinical notes all of these types of things, right? What symptoms the patient have, their comorbidities, their disease status, and so on. And doing that after the fact, after notes have been collected, is really slow and expensive. So my lab, for example, has done a lot of work on what the community calls technology-assisted abstraction, where you might use AI algorithms to try to guide annotators to extract all of these data elements from clinical notes. But it's slow. Even with AI assistance, it's slow, it's expensive, and you have the critical problem that if the information you need is not actually mentioned in the notes, then no NLP algorithm is ever going to be able to extract it. And so what we've been working on is what we call AI-driven clinical documentation, where we try to collect the data that we think is going to be needed prospectively at the point of care. We try to make it really easy for clinicians and for scribes, as invisible as possible. And as a result, we try to create semi-structured notes where we have the text that the doctors are writing, but behind the scenes, we also get structured data out. And the backdrop for this is that doctors just spend more time documenting than they do with communicating with patients. They hate their computers. Half of doctors struggle with burnout, which is directly linked to the healthcare software. And we think there's an opportunity to both collect the data we need and to address these big usability problems. So just to jump to the chase, our new user interface for documentation looks like this. On the left-hand side, you have a text box or a series of text boxes, just as existing electronic medical records do, except that as a clinician is typing in these checkboxes, we're going to be gently guiding them to help them document information that we want and get structured data at the same time. On the right-hand side, we provide some bangs for the buck. So if you're using our system, you're going to be able to also find information on patients much easier. So after documenting the patient has chest pain, we'll pull up from the patient's medical history notes that we think are useful for managing a patient with chest pain at this point in time. And in this way, reducing some of the burden that clinicians have today of having to do hundreds to thousands of clicks elsewhere in the electronic medical record to find the information that they need. So here's an example of that first part. You see that the doctor is typing in, and we're using machine learning algorithms, the same algorithms that we have on our cell phone for text messaging or Gmail, to do prediction of what, or autocomplete of what it is the clinician is likely to type in. Although from their perspective, it's just autocompleting text, behind the scenes, every single thing we're autocompleting is linked to a medical ontology. And so what we get out is the note plus all of these quantities that have been linked to structured data. In order to get that autocomplete to run in the browser, we built a small convolutional neural network language model that is still quite accurate despite its size. And we showed how one could use this data that one collects also to save clinicians time. For example, to pre-fill later parts of the note with information that has already been collected. And I already mentioned about this. So a pilot at Beth Deaconess Medical Center found very high usability scores. This was largely used by scribes in this pilot, although there were some attendings that used it as well. And some of the comments that we got were that the auto-populated text makes documentation feel more efficient, less error prone. That relevant information on the cards that I showed on the right-hand side prompted the users to document information that they might not have otherwise documented. So in conclusion, I think it's going to be very exciting to see the next few years where we are going to develop the next generation of AI-driven electronic medical records that doctors love and that also collect the right information at the point in care. And I'll conclude there. And I see there's some questions.
Thank you so much. So maybe I can raise people, I guess just information for everyone. If you have questions, raise your hand. I can raise you as a panelist, so you can directly ask your questions. There are also 2 questions typed in the chat. Xiao Liu, are you still here? Let me see if I can make you, you can also read the question.
I can read out loud the question. The question is about the second part of the talk of subtyping, and Xiao asks, does identifiability rely on the assumption that the disease sequence is weakly monotonic, so a patient cannot improve for a period and then get worse later? It's a great question. No, we're not strictly making that assumption. But we are making assumptions on the parametric form of what the trajectory of the subtype of disease progression might look like. So for example, maybe we have to assume it is a quadratic function, but it could be a quadratic function going up, could be a quadratic function going down, or could even be sort of going down and going back up. So we're not going to force monotonicity, but we are going to make some assumptions about the actual form of it, and that is needed for identifiability. I should note that there are many open questions here, and I encourage you, if you're looking for new problems to work on, to take a look at the paper and see some of those.
Thank you so much. Another question is in the Q&A, and it ended up in the answered because I put a comment on it. So it's by Tat Thongvo, which you can see the questions.
I see it now, yes. Which question are you asking about?
How can we measure it's in the answered, it ended up in the answered.
Oh, in the answered one. Okay, I get it. 1 second. Okay, I find it now. How can we measure the uncertainty, say, by constructing 95% confidence bands when predicting the future outcomes? So I think you might be asking about that first part of the talk on the patient simulator. And because of it also being a Bayesian approach where we are modeling the distribution over all of the patient's future outcomes, you can actually get, you can either do, there are a couple of ways to do it, but the simplest way to think about it is you can do many rollouts. So you can sample either many times, or you can use some probabilistic inference to marginalize and get the marginal probability of any one observation. And then with those, then you can construct confidence intervals such as the one that you're asking for. And that's actually not something that we did in our paper. It's something that's possible to do, but also something that I think would be really important for follow-up work in this direction. I encourage you to do it if you're interested in it.
And then two more questions got added in the Q&A.
Okay,
open one, Mingzhu and Elisa.
Okay, so first, Mingzhu's question: does the Sublime method assume a combinatorial model? For example, a combination of certain biomarkers would lead to a certain subphenotype cluster, or an additive model such that the presence of enough numbers of biomarkers would lead to a certain subphenotype cluster. It's more of a combinatorial nature, but with one important limitation, which is that our model assumes that the biomarkers sequences are all conditionally independent of one another, given the time points they were observed and given the Z and the delta. Z, remember, is the quantity which is describing the patient's physiological state. It's a high dimensional unobserved variable, which we're doing inference over. So we definitely are modeling the correlations between the biomarkers, but we're doing it using that conditional independence assumption. Move on to the next question from Elisa. The question is, for MedKnowts, did we curate label data, or are we falling back on search heuristics, and is there a way to demo it? So For the last question, I will just give you a link in the chat here to medknowts.org, knowts.org. And from there, you can find a longer 5-minute video, which walks you through the system as it's been designed. There isn't a way to play around with it on your own, partly because that would require us coming up with very good synthetic data to include in it. We wouldn't want to share any real patient data. But if you want to know more, I'd be happy to talk to you offline. You can just email me. With regards to your first question about curation of the labeled data, so going back to perhaps this one, so we use to build this autocomplete infrastructure. There were 2 steps to it. The first step was to take existing clinical notes. We took a few 100,000 existing clinical notes, and we used a natural language processing algorithms to try to extract clinical concepts from those existing notes. And my research group actually has just had a pretty major advance on how to do that. And I'll send a link here as well in case everyone's interested. It's work that's also done by my student Monica Agrawal on using large language models exactly for that purpose. So I just sent a link in the chat in case any of you are curious. So we would use an approach like that to first identify mentions of things that we would like to be able to autocomplete. And then we will then learn a machine learning model that will take the data up until the identified mentions and predict which of the mentions it's going to be next. And that's how we then can cold start the system so that even before getting high quality labeled data through the use of this new interface, we can still get reasonable predictions of what the clinician's likely to type next. I hope that answers your question.
Can I ask a follow-up question, David, on this? So what do you think about potential bias in the clinician in saying, okay, you know, this patient might have this thing. I didn't necessarily see it, but 90% of them, the patients with certain background have it. It's auto-completed, just like I'm in a rush. I'm just gonna like accept it and move on or something. Basically, that is a little bit of a worrisome thing where it's just like cognitive bias and people would kick in, they accept things that are just based on statistical errors.
Yeah, and I think that's something that's an extremely important question. I'll just restate your question, I guess, for everyone, which is that the moment we start guiding clinicians in documentation, you have to worry that maybe we're going to guide them in a way which is wrong or inappropriate or leads to additional biases where there weren't biases before or new biases. And indeed, that's an extremely important concern. And any deployment of these systems needs to have checks and balances for precisely that. Now, the good news is that I have done one example of such a study, and I'll send a link in the chat now. This is work that Stephen Horing and I had done many years ago for our earliest example of such an autocomplete system, which has already been used for several 100,000 patients in the emergency department. And we did a post hoc study after several years of being in deployment to understand what biases and it might have had and how the quality of the data that was collected, now that you had AI guiding the documentation, how that quality changed. And going into the study, we didn't know what to expect because we had exactly the same concerns that Nargis just raised. We were very pleasantly surprised to find that the quality of the data actually improved substantially through the system. And it's this type of study that I think is important to keep redoing. And it's part of, I think, where the academic community's focus has to be to make sure that we're actually not hurting the state of the world at the same time as we think we're making progress.
Awesome. This is exciting. Thank you. I don't know how much longer you have. Thanks for staying. It's 10 minutes past.
I suggest we maybe adjourn there, but I'd love if anyone has any questions they want to talk to me later, please let me know. And I do hope to come by NYU at a future date.
Absolutely. We would love to have you.
Right, thank you, everyone. Thank you, Nargis, for the invitation.
Thank you, David, for the amazing talk and answering the questions. Have fun